Hello students, in the previous lesson we have already learnt about the main reproductive organ, that is the stamen. There we have already learnt that how the male gametes are being formed. In today's lesson we are going to learn about the structure and function of the female reproductive organ, that is the gynoecium, its the structure of the megasporangium, and how the female gametophyte is being formed. Now this is the female reproductive part, where you are going to see that this structure, this is the stigma, this is the style, and this one is the ovary. Now the stigma is the pollen receiving site through which the pollen enters and it passes through the style and reaches the ovary. Inside this ovary, there is ovules. We are going to learn about its structure as well a little bit later. Now, the female reproductive part has got different types. First one is the monocarpellary. That means it has got one pistil or single pistil. Multicarpellary means when the gynoecium has got more than one pistil. Another type of classification can be made, that is apocarpous. Apocarpous means that is gynoecium with free pistil. Free pistil means like this one. And syncarpous means the one which is fused like this. It has got fused. Part we are going to learn about the structure of this ovules. Now the structure of a typical ovule here. Now this ovule has got two main parts. A stalk-like structure, this one, which is the funicle, and the body part which has got two layers. This one, this layer, and this layer. These two layers, which is known as integument. Now this integument can be bitegmic and unitegmic as well. In gymnosperms, it is basically unitegmic, and in angiosperms, it is basically bitegmic. Bitegmic means it has got two integuments. However, in gymnosperms, only one integument is present, and that is why it is known as unitegmic. Now, this is the central part of the ovule, and this ovule is filled. This central part is filled with mass of cells, and this is called nucellus. Now the portion that is stretched over this body part, this portion, this is called the raphe. And the place where the funicle is attached to the body, here, this is called hilum. Now, as I said, the central part is known as the nucellus. Now, this nucellus can be of two types. One is the tannoid nucellus. That means when the nucellus is of less in content, then it is said to be tannoid nucellus. Another one is known as the quasi-nucellate, where the nucellus is plenty. That means the mass of cells here is plenty. Now, this integument, where it is discontinuous at a particular point, that is when the integument leaves a passage here, this passage is known as the micropyle, and the site of micropyle, just the opposite side of the micropyle, is the chalaza. This is the chalaza region. Now, when the ovule is being developed, what happens? The nucellus means it has got mass of cells. When one of the cells towards this micropyle, they become bigger, then they are said to be the megaspore mother cell. This one is the megaspore mother cell. And this integument that we are talking about, this will ultimately turn into seed coat, and the entire structure, will eventually change to seed. In the next part, we are going to learn about the different types of ovule. In the previous part, we have already learned about the structure of the ovule. Here we are going to learn about the different types of ovule. There are basically 6 different types. The first one is the orthotropous. Ortho means straight and tropous means turned. Now if we are already aware of the structures. Now these are integuments. This is the embryo sac. This one is the nucellus, we have already learned, and this part is the chalaza. Now when this micropyle, the chalaza and the funicle, they lie in one straight line here, then that is known as orthotropous, and this is found in the polygonaceae or nucleaceae. Next type of ovule that we get to see is the anatropous. Ana means backward and tropous means turned. Now if you see the structure, this one is the chalaza, this one is the nucellus, this is the embryo sac, these are the integuments, this is the micropyle, and this part is the funicle. Now in anatropous ovule, here the body of the ovule turns backward by an angle of 180 degree, and so the micropyle becomes very close to the hilum and the placenta, and this is found in the sympetally. Next type of ovule that we see is the hemitropous. Now hemi means half and tropous means turned. If we see the structure here in hemitropous, then this one is the chalaza, this one is the nucellus, this one is the embryo sac, and this part is the micropyle, and this part is the funicle. Now in hemitropous, the body of this ovule is placed transversely here. You can say somewhat at right angle to the funicle, like this. So this is the funicle, this is at right angle, like this. And here the chalaza and the micropyle that are present is in one straight line, and this is found in the ranunculus. Next type of ovule that we get to see is the campylotropous. Campylos means curved and tropous means turned. If you see the structure here, this one is the micropyle, this one is the embryo sac, this is the funicle, and this is the nucellus. Here, the body of the ovule is curved in such a way that the chalaza and the micropyle, they do not lie in the same straight line, and this is found in the leguminosae family. If we see the structure of the amphitropous, we will see that this part is the micropyle, this is the embryo sac, this is the nucellus, and these are the integuments. Okay, and this part is the funicle. Here what happens, the curvature of the ovule here is very prominent or rather pronounced, and the embryo sac here, it becomes somewhat like a horseshoe shaped, and this is found basically in butomaceae. Next type of ovule that we get to see is the sarcinotropous. In sarcinotropous, now if we say this is the embryo sac, this one is the chalaza, this part is the micropyle, and this part is the funicle. Now this funicle, here is very long, and the ovule here rotates by an angle of 360 degrees, and it is put in such a manner that it is completely circled around like this. It is completely circled around by the funicle. Here the micropyle faces upwards, and this is found in the Cactaceae family. In the next part, we are going to see how the megaspore mother cell that we have already discussed when we were discussing the structure of a typical ovule, how this MMC changes into megaspore and how the female gametophyte is formed. Now we are going to see the development of the female gametophyte, that is the embryo sac. We have already learned about the different types of ovule. However, the most common type that we study for the development of the female gametophyte is the anatropous ovule here. Here, we have already learned about the different types of structure of the ovule of the anatropous ovule runner. But our main focus for the development of female gametophyte is the nucellus, this mass of cells that is present in the central part. Well, what happens when one cell towards the micropyle, they change to MMC, that is the megaspore mother cell. And also we need to know the location of the two ends. This end, that is the integument, that becomes discontinuous in a part of the ovule. That part is known as the micropyle, the passage. This passage, and just the opposite of micropyle is the chalaza region. Now, in the next part, we are going to see that when the ovules differentiate and form one megaspore mother cell, what really happens? Now, the formation of the megaspore is termed as megasporogenesis. And this is basically formed in the megasporangium. Now what happens? Firstly, ovules differentiate to form one megaspore mother cell. Now this megaspore mother cell undergoes meiosis, the first meiotic division. The first meiotic division results in the formation of 4 haploid megaspores, and these megaspores are known as tetrad. This we have already learned when we have studied the male gametogenesis. In the same way, here also the megaspore mother cell, it undergoes the first meiotic division and it results into the formation of megaspore tetrad formation. In the androecium, what happens? In the structure of the stamen, what happens? This tetrad formation can be of different patterns. We have already discussed that. But in case of megasporogenesis, here only one type of pattern is observed, that is the linear pattern. Okay. Now, out of this 4 megaspores, one of these megaspores, that is monosporic, is at the chalazal end, that develops into female gametophyte, and the other three megaspores which are near the micropylar end, they get degenerated. The one which becomes functional eventually, that is a functional megaspore which is haploid in nature, this enlarges, and as it develops, it results into the formation of the female gametophyte. Now this functional megaspore is the first cell of the female gametophyte. The functional megaspore that we have discussed here, it undergoes certain divisions. Now, it is going to first undergo karyokinesis, that is the division of nucleus, and this karyokinesis occurs 3 times. The nucleus of the embryo sac, that divides into 2 and then 4 and then what happens? 8 daughter nuclei, 4 of which are located at each pole. Now the 3 nuclear minority divisions that occur, that means the nucleus of the embryo first divides into 2, then it divides into 4, and then finally it divides into 8 daughter nuclei. And these are located at each poles. And this nuclei, what happens? They rearrange themselves. How? 3 of them, all total 8 daughter nuclei are being formed. 3 that are being formed, they are making their position at the chalaza region, and 3 moves towards the micropyle region, and the 2 remains in the central part. Now this is followed by cytokinesis, that is the division of cytoplasm. What happens then? 3 cells towards the chalaza region. This region, 3 cells towards the chalaza region, they results in the formation of antipodal cells, this 3. And the 3 in the micropyle region, they form the egg apparatus. This full part is the egg apparatus. Now, this egg apparatus consists of what? The 2 flask-shaped synergies like this, and a round cell or a round egg cell, that is the ovum or the oospore, that hangs between these 2 synergies, and the rest, the 2 cells that remains at the center, they remain as it is. This embryo sac that is being formed in the angiosperm is basically 8 nucleated. That means 8 daughter, because why? 8 daughter nuclei after 3 nuclear minority divisions, that means 8 nucleated, and it has got 7 cells. How 7 cells? This 1, 2, 3, 4, 5, 6, and this one is 7. So 7 cells. And this type of arrangement is known as the polygonum type embryo sac. This is how the female gametophyte is being formed. In the next part, we are going to see how the male gamete that has been formed is being transferred from the anther to the female reproductive part, that is the stigma. This process is known as pollination.